So the project that we started a few years ago, essentially we needed to show that we could get these into animals and not cause side effects. I've already told you a little bit about the physics. We've done some nice work on the physics of this. We had to show that we could kill cancer cells, both in a dish and tumors that are malignant tumors in an animal. We also had to find out if there were any side effects to doing this. You know, the old saying is there's no free lunch. We know that if we do too much damage to these cancer cells or if we damage too much tissue, we may cause side effects. We needed to find that out. We also needed to know, and this is really the so what question, can we add other substances, antibodies, proteins, drugs, to the nanoparticles to get them preferentially taken up by the cancer cell? That's what we know we need to do. We need these taken up by the cancer cell, and we don't want to get them in any normal cells because if we do, the normal cells will be damaged. And we're looking at a broad array. Again, those of you who last heard me talk, we really were concentrating only on these first three. In the last year, since I've been here, we've expanded to a number of other tumor cell types, as you can see, breast cancer, types of sarcomas, skin cancer, lymphomas, leukemias. These are particularly fascinating to me. One, because my good friend John Canzius is dealing with leukemia right now, but two, we weren't sure we could deal with the so-called liquid tumors, tumors that circulate. We now have very good data to suggest that we can. Well, how do we prove that we're killing these cells? And I'll have to teach you a little bit about some of the tools we use in research. There's a type of a study called a flow cytometer. And what a flow cytometer tells you is if you're doing a treatment that is actually damaging or killing cells. And we use a couple of fluorescent types of molecules. One is a molecule called propydium iodide, and what it's telling you is, are your cells alive and going through the normal cell cycle, meaning they're alive and they're happy and they divide and one cell goes into two, etc. Well, a normal flow histogram looks like this, meaning that most of your cells are in this phase, so-called G0. And that's the phase where cells are doing the things that cells in that particular organ do. If they're liver cells, they're making bile and they're detoxifying drugs and they're handling all the food that goes through your intestine. Some cells are actually going through, making new DNA, and other cells are actually getting ready to divide into two cells. So what we then did is we looked at that technique, and we then took cancer cells and we put them in a dish and we put them in the RF field. And here's an example of that. So here's a dish with some cancer cells, and you see we've got it on this Teflon plate. Notice that there's nothing touching the cells, nothing touching the plate. It's on just this Teflon holder. And again, we've learned we have to use things like Teflon. We can't put metal things in there because it would arc across there. But essentially, again, you turn on the machine and you don't see anything or feel anything happening. You're just kind of hoping, gee, I hope this is doing something. Well, here's an example of a pancreas cancer cell. Pancreas cancer, as some of you may know, is a particularly evil cancer we deal with. I unfortunately have chosen as an interest cells that are particularly tough to treat. Pancreas cancer, liver cancer, some types of colon cancer. Many patients, I'm sorry to say, and frankly the vast majority of patients who get pancreas cancer die as a result of this cancer. We do not have good treatments for this type of cancer. We need to do better. So this is one that I studied early on. So this is an example of a pancreas cancer cell it's a human cell line. And we treated it, no nanoparticles, and we put it in the radiofrequency field, and here again you see normal cells. So this is absolutely like you did nothing to them, proving that the radiofrequency field itself doesn't do anything bad to the cells. Now if you add gold nanoparticles, notice this is all shifted over here. These are all essentially cells that have been literally blown apart. They're just fragments of cells. There are no live cells left. They've been 100% killed by this treatment. And this was actually a little bit longer than we needed to do, but this is when we were still in kind of a learning curve. Now here's an example. Remember I showed you some electron microscopy before, and I know a lot of you probably have never looked at things like that, but here's a cell that, again, we treated with the gold nanoparticles, and it's a pancreas cancer cell, but no radiofrequency treatment. Now this is a normal cell. It just happens to have these gold in it. So there's normal Golgi apparatus, normal endoplasmic reticulum, the membrane, everything is intact. Here's a cell treated with the RF. Notice there's lots of spaces here. It looks airy. We've literally blown this cell apart. You'll notice there's stuff leaking out of the cell membrane, literally. We've destroyed the cell membrane. We've coagulated proteins in it. This is a dead cell by anybody's criteria. We showed several of these photomicrographs to people who had no idea what we'd done to the cells. First of all, they said, what the heck is this stuff, and what the heck did you guys do to this cell? It's totally blown to pieces. So this proved that we could actually get the nanoparticles into the cells, expose them to the radiofrequency field, and kill the cells completely. Here's an example now using the carbon nanotubes, and this is with a liver cancer cell line. Again, cells treated with just RF alone or with the carbon nanotubes. Again, you see complete killing of the cells. Another type of liver cancer cell line, complete killing of the cells. And finally, pancreatic cancer cells, again now with the carbon nanotubes instead of the gold, almost complete killing. And what we found is the carbon nanotubes did not get into the pancreas cells in as high as levels as the gold. So again, the gold here was seen to have an advantage. Well, we realized in doing this that there was actually dose response and that we could get doses. In this case, this correlates to a very low dose, 50 milligrams per liter. That sounds like a lot, but that's actually a very small dose in terms of a liter distribution. And, 500. And we were able, again, to get complete killing of all three types of cancer cells when we went to that high a concentration. Well, that's great. I mean, unfortunately, many of you have seen things in Time magazine, Newsweek magazine, examples of some great cancer treatment, some new cure, and it's usually been based on somebody killing cancer cells in a dish or in some experimental animals. I intentionally don't use experimental animals that are highly inbred because we are trying to show that we can kill tumors in a population that are more similar to humans who are not an inbred population. So we have a type of a rabbit model that grows tumors, malignant tumors in their liver. This is called a VX2 tumor. Essentially what we did is, and this is with the carbon nanotubes, we had the tumor growing in the liver, we injected some of these carbon nanotubes directly into that tumor, and then we treated the animal with only 2 minutes of radiofrequency ablation. What we found is if we put a little temperature probe right next to the tumor, during the treatment, the temperature steadily rose up to about 55 to 60 degrees centigrade. Again, 37 degrees centigrade is normal body temperature. However, thermistor is just a few centimeters away, no heating at all. Again, showing we're only getting heating in the tissue that had the nanotubes in them. When we were able to then take these tumors out and look at them, we found that we got complete killing, whereas tumors that were not injected with nanotubes, that just got a salt solution, were completely live. And what does that look like? Well, here's an example of one of the tumors that was treated with the nanoparticles. And again, you can see all the coagulated black stuff here. These are the carbon nanotubes that have literally come out of solution and clumped together after the RF treatment. And what you're seeing here is essentially, under a higher power, nothing but inflammatory cells. I know that most of you have never probably spent a lot of time looking at cancers or tumors under a microscope. What you're seeing here is just a lot of what we call necrotic or dead debris. Inflammatory cells, cells that are coming in from the body's immune system to kind of clean up this response. If we stain for this, what we're finding is what we call neutrophils or macrophages in this field. When we did a stain looking for the tumor cells, we found none. Here's an example of one of the control tumors. Now, this is a normal VX2 tumor, again under a higher power field. And this is what VX2 tumors that are growing normally look like. And you can see that's pretty much you don't see all the debris, you don't see all those weird little cells, the inflammatory cells. So again, proving that if you just treat the tumor with RF, John's RF field alone, doesn't hurt the animal, doesn't hurt the liver, unfortunately, doesn't hurt the tumor. You've got to have the nanoparticles there. Well, John will tell you I'm a bit of a nut for these things. I have to prove it to myself not one way, but usually 2 or 3 ways. So there are assays that are looking at what we call apoptosis, cells that are dead or dying. This is what's called a tunnel assay. This is a type of stain looking for killing of the cancer cells. And what this stain shows is that a cell that is dead is brown. Cells that are alive are blue. So any tumor has maybe 1 or 2% of the cancer cells that are dead. These tumors are undergoing the processes. They sometimes grow rapidly. So I remember I told you brown is dead. So here's the tumor treated with the carbon nanotubes. A lot of brown in there. And again, so all of this debris has been stained. And again, when we counterstain, these are all essentially either normal liver cells in the area or debris or, again, the macrophages.